Welcome to Annual Research Review 2024 NIFS. I am Dhammika Magana-Arachchi, leader for the research program Molecular Microbiology and Human Diseases. Our research revolves around microbes, their interactions with the environment, and also we are working on human diseases, both communicable and uncommunicable diseases. Last year, as listed in action plan, we conducted our research based on 4 areas: human diseases, environment, cyanobacteria and cyanotoxins, and screening microbes for commercial utility under 10 sub-projects. Here I will present only selected research with the time. In first sub-project, transcriptomic analysis of mycobacteria in serum exosomes of latent tuberculosis patients for candidate biomarker identification funded by NIFS. We have completed this project and we identified human-derived differentially expressed genes in both whole blood and also exosomes. However, pathogen-derived differentially expressed genes were identified only in serum exosomes of latent tuberculosis individuals. This can be used as a potential biomarker for molecular diagnostic test for tuberculosis. And we are also looking at the gene expressions of serum of host, that is human expression, to identify potential biomarkers for latent TB. In this sub-project, enrichment mechanism of CKDU risk factors in groundwater, their uptake pathways and potential remedies funded by NSF, we completed this project, and important findings included around 93% of samples exceeded the WHO guidelines value of 1 microgram per liter of microcystins in drinking water. Also, cylindrospomopsin mean concentration in CKDU well waters was approximately 5-fold higher compared to the non-endemic areas. In this sub-project, to identify urinary biomarkers and urine microbiome for diabetic and hypertensive CKD in Sri Lanka, we have completed this project also. We identified a panel of genes which would be valuable for developing a comprehensive biomarker profile for diabetic nephropathy. Will aid in timely diagnosis. And also we identified a gene which could be used as a biomarker for hypertensive nephropathy. In the sub-project, determining the microorganism diversity at high altitudes, we compared the airborne bacterial diversity across contrasting altitudes. And we were able to collect samples from Twidrutalagala mountain top as well as from Lotus Tower, 2 different geographical areas. And here listed the dominant bacterial genera identified in both locations. Interestingly, several organisms recorded from our study have been previously documented in high altitude environmental research, such as Bacillus altitudinis and also Bacillus aerophilus. The sub-project on balloon flights over central Sri Lanka, the objective was to establish that the Earth is continuously receiving microorganisms from space, which contribute to the evolution of life on our planet. The concept of Professor Chandra Wickramasinghe. Project was initiated in 2017, but we had to terminate this project due to the unavailability of permission from the Met Department and also from the Civil Aviation Authority of Sri Lanka, because without their permission, we cannot launch a balloon. Here, we monitor the transboundary haze events, and we compared the microbial load and diversity during haze events in Kandy. When compared to the microbial load in 2022, the microbial load we observed in 2024 was 18 times higher. And also, evenings consistently showed significantly higher microbial levels compared to morning and noon. We have also screening and try to identify bacteria which could be used in industry, and we have successfully isolated certain bacteria which are potential for the enzyme in industry. In this project, we are determining the variability of microcystin producing gene cluster in selected cyanobacterial isolates, because the microcystin production is correlated with the presence of MYC gene. Here, the average toxin concentration showed a significant positive correlation with the presence of selected MYC gene percentage. And last year, I supervised students, PhDs, MPhils, and also undergraduates. And I am happy to say that one of my students was able to complete her PhD less than 4 years, which is an achievement in Sri Lanka. My research impact, actually the research group impact, can be categorized under academic, educational, economic, health and technology, environmental, and even for policymakers. These are my group members, project members, and also I wish to thank my collaborators, supervisors, funding bodies, and NIFS, past and present directors, and also to the staff. Thank you.